Hello and welcome to a brand new session today on the topic structure of Microsporangium in flowering plants. Yeah, this is a topic from the chapter Sexual Reproduction in Flowering Plants picked from the NCERT syllabus of class 12th. It's the second chapter probably. So today we will be going through deeply about the concept structure of Microsporangium. We will be going through all the layers, all the parts, what is Tetrasporangiate, what is Dithecus. We'll be reading between the lines. So get ready for a wonderful session. Yeah, welcome. Are you excited for a fun learning session with me? Yes, you better be. You will only be excited by the way I teach. So no need to worry about that. Just be concentrated and just be always attentive so that you grasp important things and important concepts out there. Yeah, as already mentioned, the agenda for today's class is structure of Microsporangium. What is Microsporangium? What is inside that? What are the parts of it? What are the layers present? What is the function of the layers? Everything will be taught in detail. So please continue watching this. Yeah, this is the step. Before going directly to the concept of Microsporangium and the complex concepts, let's go through some common and basic prerequisites required for it. Yeah, we all know that in a sexually reproducing plant, angiosperms, in the flowers there will be two parts, male reproductive whorl and female reproductive whorl. Yeah, in the male reproductive part is called stamen or androecium, and the female is called pistil or gynoecium respectively. In this stamen, the stamen is made up of two parts mainly. One is anther and the other is filament. The anther is a bulb-like structure present on the top of the filament. The filament is a stalk-like structure which connects the thalamus. Thalamus is the platform on the flower. You may be knowing that. Yeah, the thalamus is connected to the anther via the filament, which is a stalk-like structure which holds the anther, which is a bulb-like structure. You can see in the picture too. Yeah, this is the filament and this is the anther. The anther is clearly seen by you, the powdery like structures, the two parts is being visible here. Just keep on observing the pictures so that you get an idea of what is happening, so that when the thing is taught for you or the thing is told for you, so that you can grab that nicely and firmly and it stays in your mind forever. So I think the stamen part is clear right now. Next, if we go to that bulb-like structure which was present on the top of the filament, it is called as anther, the terminal generally bilobed structure. Yeah, this is how the definitions are given in the NCERT textbook. So we should be ready and we should always read between the lines to understand the concept really well. The definitions are not needed in neat perspective, but for boards, these are definitely important definitions. So anther, the terminal, why is it used? Since this is the filament, we already saw what is the filament. It is present at the terminal position. Terminal position is the topmost or the ending position. It is referred to as terminal, generally bilobed. This I will be coming to this in the next slide. Hopefully, yeah. In the next slide, I will be telling what is bilobed and why is the anther called bilobed and all. Bilobe structure is called the anther. For now, let this definition be crystal clear for you once again with me. The terminal generally bilobed structure is called the anther. Terminal, why is terminal? Since it's present on the terminal position of filament. And bilobed, I have given some introduction, but it will be taken deeply in the next slide in a few minutes. So until now, what is a stamen? What does the stamen consist of? And what is the anther? And what does it consist of? It's the the basement is set right now. Now we'll be going to the important slide of our class. Yeah, this will be the remainder of the session, and what this is a holy slide actually to understand everything related to this microsporangium. I'll be marking some annotations right now, so kindly be attentive right now. Firstly, we saw that the anther is bilobed. We know that bi means two. With me on this? Yeah, bi means two. Bilobed. When we see the word bilobed, the main thing that comes to our mind is there should be two lobes. That is what exactly happens here. Normally people get confused. See, I first tell how people are confused so that you will get an idea of what not to confuse for, and the concept will be deeply set in your mind. People get oftenly confused for these round structures as lobes, and they tell that this is one lobe, this is two, and this is three, and this is four, and this Anther has four lobes, but it is referred to as bilobed, how it is and all. So it's exactly not like that. Mainly the Anther has two lobes. I'll be showing what are the two lobes. Yeah, let's erase everything for now. Two lobes are, we have seen a connective here. This connective I'll be coming to this a bit later, but firstly, what are the two lobes present? Now, since we have seen the word bilobed, the term bilobed in the definition, we know clearly that there must be two lobes. So what are the two lobes? How are they differentiated? Yeah, that is the next agenda. This part, the right part of the connective and the left part of the connective is almost identical, and these are the two lobes present in the Anther. Once again, with me on this. This is a really important concept, children. This exactly needs a great attention, and people are all most of the times confused in this part. The two lobes are present on the either sides of the connective tissue present in between. And firstly, I forgot saying this, and this is the 3D transverse section of the Anther which I have taken here. A typical 3D cross section. This I had to mention previously but didn't. I am saying this now. So let us come back to the discussion right now. What we were saying is the two lobes. How are they present? This is a small connective present here, and the two lobes are present on the either sides of it. So now you people are ready to say that these four are not the lobes. The lobes are present on the either side of the connective or this imaginary axis passing through the connective, and the parts present on the either sides which are identical, which contains two round these structures. I'll be coming to what these round structures are and what these small cells and all are. But still, till that, what you have to be crystal clear about is the Anther is bilobed. It means two lobes are present, and the two lobes are, one is this one and one is this one. So now I'm stressing upon the point that bilobed, what the lobes are and all, since it is a very important thing, and it provides a basic platform for your further understanding about the structure of an Anther. So yeah, this is what I had for bilobe structure explanation. Now you are clear about that the Anther is bilobed, and you are clear that what these two lobes are. Now we come across another term in the textbook or any books we read, that the Anther is dithecus. Yeah, again dithecus. Dithecus. If you split this word, guys, it is di. Di means, once again, two. That means theca. Theca in biological terms is referred to for a lobe. Actually, the touchpad is not present, so I'm typing via the finger itself, so the handwriting is becoming shabby. Kindly adjust with that. So, dithecus, with me on this. Yeah, bilobed and dithecus. These are actually synonyms itself for the same thing. It is the both are stressing upon. Bilobed and dithecus. Dithecus, theca is referred to as lobes. Since we have two lobes here, we say that the Anther is bilobed, and then dithecus. Clear on this? Dithecus, di means two, theca means lobe. So we are crystal clear about the thing that the Anther is bilobed and dithecus. It is referred to as bilobed because it has two lobes. Theca means lobes, and dithecus is sorted for us. Now we are sorted by two terms which we refer for an Anther is bilobed and dithecus. Now the next thing we often come across is the word is Tetrasporangiate. People often are really scared of this word. Actually, I was too. But once you understand this, it's really a cakewalk actually. Tetrasporangiate. Tetra, this word means four. We know that. Then what is this sporangiate? What is this sporangia? Sporangia must be present in 4 in number, so that the anther is called Tetrasporangiate. So now let us see what are the 4 sporangium present and what does the sporangium function is and everything. So now the sporangium, the 4 round structures which I was talking about here, which contains layers of cells, this is known as the sporangia. Sporangium. Yeah, let's recall something from the 11th class syllabus. We had seen in some division the formation of sporangium, horsetail like structures, accumulation of more spores, strobili, connecting the dots, striking the mind. Which division was it, children? Come on. Yeah, it was Pteridophyta. Pteridophyta or ferns. Yeah, we have done that. We have studied that in class 11th. But see how things come handy if we are stable, if we are strong with our 11th class concepts. Yeah, that is not required in the discussion right now, but still I just recalled about the fact that sporangium present in horse type structures, the horse tail type structures, strobili, cones. Remember everything, children? The cone-like structures which were present on pteridophytes, which contained many spores, clusters of spores. Yeah, that was a discussion for pteridophytes, strobili and all. So now let's come back to the topic here. Once again, Tetrasporangiate. 4 sporangium is present in the cross section of the anther. Yeah, we have seen here, this is 1, this is 2, this is 3, and this is 4. Going very slow on this, since these are very important concepts regarding the structure of anther, which people oftenly confuse about. So yeah, there are 4 lobes. Sorry. See, I am once again confused about it. 2 lobes, which gives the name bilobed and dithecus for the anther, and there are 4 sporangium. Sporangium is the singular noun. Actually, sporangia is the plural. Since they are present 4 in number, we refer to as sporangia. So these sporangia produce spores. Clear on this? This will be deeply taken in the next lecture, which is about microsporogenesis. Sporogenesis. Yeah, it will be taken deeply about it. What is sporangiate, how it produces spores, how the spores transform into gametes. Yeah, just and all you will be seeing in the next session. But for this session, what you have to know is these round structures which contains layers of cells is known as sporangium, and since they are present 4 in number, the anther is referred to as tetrasporangiate. Now we are clear about the two deadly or main terms about anther which we often come across, that is tetrasporangiate, bilobed, dithecus. Clear on this? With me on this? Yeah, you better be. And now let's clear that for further discussions. Yeah, now I'll be going further up about what the layers present, what are the layers present, what are their functions. Just we'll go for a glance about it, since questions are asked about it frequently. The outermost layer, let's go for a separate diagram out here. The outermost layer is present, and then another inner to it another layer is present. This is different layer. Just to remark that it is a different layer, I have marked like that. And then series of layers are present here. After this second layer, series of layers are present. This is 1, this is 2, this is third, and after that a nice layer, a nice innermost layer of covering is present here. So just I took the separate diagram for your further understanding. Now we came to know that there are 4 types of layers, from outermost to innermost, from outer to inner. With me, children, from outer to inner. From outer, the outermost layer present is known as epidermis. This word must ring a bell in your heads, children, since the epidermis we have learned many times in the ground tissue, in the tissue present in the cortex and all, in the anatomy of flowering plants chapter which we have studied in class 11. See how concepts are correlated. If you are stable with class 11 concepts, then 12th class will go like this. So now we have seen the first outermost layer, and then the second layer is present, which is known as endothelium. Endo, I'll write separately, children, with a purpose. Endothelium. Endo means inner. We have seen many times in the germ layers too, and in our female reproductive system too, that endometrium, myometrium, perimetrium. Those and all discussions will come handy in human reproduction too. But now, let's be clear on these, children. Endothelium is the next layer after epidermis. From outermost, it is the second layer. From outer to inner. With me, children. From outer, epidermis, endothelium. Clear on this? Yeah, endothelium is clear. Then we'll go dig deep inside and see these layers of fiber like things. This is known as middle layers. Middle layers. What these middle layers, in the middle layers concept, what is important for your NEET and board's perspective is, what is its function? The function is, I'll be taking this side of the PPT, I mean the left top corner. Yeah, the function is, it has alpha cellulose fibers in it. Sorry, my handwriting is going really shabby out here. Oh god. Yeah, I'll take the ink. Alpha cellulose. Yeah, what is the importance of presence of alpha cellulose in it? Yeah, it helps in absorbing water and becoming inflating, and then it helps in dispersion of the pollen grains present inside. That cannot be written, guys. Listen to me. The alpha cellulose fibers present in the middle layers absorb water. The absorbing of water develops a turgor pressure inside, which in turn helps in dispersion of the pollen grains via the stomium or the line of dehiscence. Stomium and line of dehiscence may seem foreign for you guys, but it will be discussed further. Just I am giving a teaser of it here. So you are clear with the function of middle layers right now? I'm going very slow. Revision is the key, children. Revision is the only key for success. Everyone will cover the syllabus once or twice, but the thing which decides, which segregates one from a good ranker and an average ranker is what and how much times he revises each concept. This must be kept in mind. Revision is an important asset in our preparation. Any exam may be, but revision is always an important key. So let's come back to our discussions. So now we are done with 3 layers, from outside to inside. We are singing the song, children. Outside to inside. Epidermis, endothelium, middle layers, and there comes a next layer which is not yet discussed. It is the most important layer actually, the tapatum. Sounds really exotic, right? Yeah, tapatum. Tapatum is the innermost layer which is in directly contact with our what we have discussed as a Tetrasporangiate. The sporangium, what it is present in 4 in number. Yeah, that is what covers in, or what is in close contact with the tapatum. Innermost layer from outside, but the closest present or the nearest neighbor for sporangium. Yeah, we see the nearest neighbor terms in the solid state chapter in chemistry, where we use that to find coordination number. Yeah, let it be aside. Nearest, I mean the tapetum is in close contact with the sporangium, which helps it and gives it an important function of nourishment. I'm going very slow because these are really important concepts and people are often extremely confused. Even I was confused about it when I read it for the first time. So yeah, now let's revise, children. 2 lobes, dithecus, bilobed, 4 sporangium. Sporangia is a plural one. and this gives it the name Tetrasporangiate. And then now we are seeing about the anatomy of the layers present. The outermost is epidermis. The second after it is endothelium. Thesium, theca. Here we already know, children. Others may be confused about the term theca, but we know that theca represent a lobe. Feeling happy right now, right? Yeah, we are confident in the word theca that it is a lobe. With me on this? Yeah. Epidermis, endothelium, middle layers, and then comes the tapatum. Tapatum acts like a mother layer. Yeah, this may be a mnemonic for remembering. We always compare mother, we always, sorry, the comparing becomes a wrong word here. Technically, we always refer to moms as taking care of us, nourishing us, taking extreme care of us in these tough times and all. So I always remember the same way. I always assign tapetum for mother, since how moms nourish their children. The same way the tapetum layer nourishes the developing pollen grains inside the sporangium. Yeah, these words may seem foreign for you, but it will be crystal clear in the next lecture, where I will be taking about the microsporogenesis which occurs and which results in pollen grains, a vegetative cell, a generative cell. Yeah, these and all are complex concepts, but it will be made quite easy by me in the next sessions. So stay tuned so that you may get them soon itself. So yeah, now we are clear about the layers, about the terms regarding the anther. So let's revise for one more time, children. It will be better if we do thrice. Epidermis, endothelium, middle layers, tapetum, middle layers. For you, middle layers function should be a cakewalk, children. Every time you remember, you hear somewhere middle layers, then alpha cellulose fibers must hit your mind, and they should always be shouting the function of middle layers. It is helping in dispersion of pollen grains. Yeah, connective. I have already given a teaser about it while I was discussing about the location of lobes. Connective is a vascular blend. It acts like a gum. It holds two lobes together, or else if this connective was not there, then the lobes would have been separated or segregated from each other. So connective, it's not a big thing. It just holds the lobes together. But yeah, then when I was discussing about this alpha cellulose, it struck in my mind one of the important concepts here. What is this? I mean, yeah, I'll be clearing everything so that you may get a clear perspective of what I am delivering and what I am trying to say to you guys. This region, attention children, this is one of the most important concepts of our class. Questions are there regarding this. This region is present in both the lobes, in between the sporangium. Sporangium word doesn't never scare us from today. We know what is a sporangium. We know why anther is called a tetrasporangiate. I am reminding many times, since it is an important asset of this anther structure. Yeah, this what I have marked here. This region is where the pollen grains come out. Pollen grains from junior classes, you may have some idea, children, about that they contain the male gametes, but it will be, I'm repeating it, it will be crystal clear in the next lecture, where I deeply discuss microsporogenesis. For now, this region is known as stomium, which refers to mouth like opening, but here this region refers to stomium, and pollen grains are dispersed via this, and this line through which the pollen grains get dispersed. But this 2D representation cannot help me in delivering what I am trying to deliver to you. Yeah, in cross product and all, you may know that let it this be vector A, this be vector B, then from right hand thumb rule, then if we go like this, from A cross B is pointing towards me, I mean towards you from the screen it is coming towards you, the direction. We use a dot for it, and for B cross A we use cross for it. Yeah, what I'm trying to say is the direction. This is the transverse section. If we watch it from right side, I mean the funicle. Yeah, let me try here and go to the previous slides, whether I can get a good yeah. Here I am getting a clear thing about what I am trying to reach out to you. Yeah. Once again, children, stomium is this region, but the line of dehiscence extends throughout the length of the anther. Seeing here what I said in the previous slide, this is the stomium, and this stomium extends along the length of the anther, which is known as line of dehiscence. Clear on this? Once again, with me, children. The line of dehiscence is the thing which extends throughout the length of the anther, which is present on both the lobes in between two sporangiums present in each lobe. This helps in dehiscence. Dehiscence means anthers coming out of pollen grains coming out of anther. Now my children are crystal clear about what all are present in the structure of an anther. But one topic is just left. Yeah, recall meiosis. Let's recall some concepts from 11th again. Recall meiosis. Meiosis, where we saw a 2n cell giving rise to 4n cells. Recall the reductional division which we discussed. This happens in gametogenesis, zygotic meiosis. Yeah, many things are there, children. You should or else you must be very strong in your 11th concept, so that the 12th concepts go like this. Yeah, 12th concepts become an easy peasy work when you are strong with your 11th concepts. Yeah, this is just a reductional division where 2n cell on division gives 4 haploid cells. Yeah, haploid and diploid, John wanted to confuse you on those terms here, where we are going very smooth and very slow and very accurate on what we have to learn and what we have learned. Yeah, the cell which undergoes meiosis is known as myocyte. This is known as myocyte, and these are called daughter cells. Yeah, this we know from binary fission too. Now, what is the link of meiosis and the structure of anther? You may be asking, and your mind may be striking that many times and ringing the bell many times. Excuse me. Yeah. Sporogenous cells. These sporogenous cells are the ones which undergo meiosis to produce the pollen grain, or what we see as pollen grains, not generally pollen grains in the parasite itself. It produces spores, microspores. 4 of them are produced. There is many thing about it which will be once again telling in the next lecture. Next lecture you will be extremely done with all the concepts regarding this, and you will be good to go. So now, once again, what we are doing now? We are doing sporogenous tissue. Sporogenous tissue is the thing which is present inside the tapatum. Inside the tapatum means in the Tetrasporangiate, the 4 sporangium which we learned, the sporogenous tissue is present in the sporangium. Yeah, we all know now that these sporogenous cells here, these sporogenous cells undergo meiosis and help somehow. For now, let's know that somehow sporogenous cells undergo meiosis and help in the production of that what we see as pollen grains. But now we are clear about this, children, that sporogenous tissue is present in sporangium, and what is the function of it? It helps in gamete production or pollen grain production or gametophyte production. Okay, now that you are crystal clear about all the concepts regarding this, what all we have learned today, children? The bilobed and the dithecous anther, which contains 2 lobes, What are the two lobes? I have clearly demarked everything and we have dissected the concept to a maximum extent. And then we came to the discussion of what is a sporangium, what is the function of it, how many are present in number, and what it does, and why the anther is known as Tetrasporangiate. So many concepts have been learned today. Congratulations, children. Next, we went on to the anatomy of the layers present from outside to inside. From outside to inside. We learned that from outside we have epidermis, endothelium, middle layers, and tapatum. We will be going through important functions here. So we saw the function of middle layers. Ringing the bell, children. You must ring the bell. This is an important thing. I have repeated twice or thrice, I think. Middle layers contain alpha cellulose fibers, which in turn help absorb water, and hygroscopic pressure is developed. Yeah, that will be done. I'll do one thing. In the next lecture, I'll be going a bit deep on it. For now, it's enough that if you remember the function of what middle layers does, what it consists of, it is enough. Then if we go inside the tapatum, what I gave the analogy of mother, mom, tapatum, everything are the same. Mom nourishes us, mom helps us in development. In the same way, tapatum helps the sporangium, tapatum helps the spores growing in it, tapatum helps the pollen grains growing in it. Yeah, tapatum nourishes the gametophyte or the sporophyte which is developing inside the sporangium. This is what you have to note today. And then connective is not a big concept. I mean, I have told already that it is a vascular blend of cells which hold the two lobes together. And we went to learn about one of the important concepts of today, the line of dehiscence, stomium, which is the exit point for all the pollen grains. Pollen grains must be dispersed, children. It doesn't have any use if it is hidden inside the sporogenous tissue, inside the sporangium. So line of dehiscence, stomium was clear for you. And then at last I came back to sporogenous tissue which is present inside this sporangium, which plays a major role in undergoing meiosis and producing spores, haploid spores. Yeah, that and all you will be learning deeply, children. Don't need to worry about it. So now you are sorted, children. Congratulations on getting each and every concept crystal clear for your mind so that you never forget this. Ensure revising this as many times as you can, since revision is the only key for success and it segregates the people from becoming a top rankers and an average rankers. I've been saying that since I have experienced how revision helps and what happens if we don't revise concepts regularly. Now let's move on to the next. Yeah, how much have you learned? Assessment time, question time. Ready for the question? How do the pollen grains break open from the pollen sacs? Yeah, how do the pollen grains break open from the pollen sacs? Yeah, children, you must answer this. You should get this in before 5 seconds itself. Since you have learned each and every part of the anther crystal clearly, 3 times, 4 times we have done it, children. You must get the answer. The answer is, the correct answer is line of dehiscence. Connective, we know that it's a vascular blend which holds both the lobes. And then line of dehiscence we already saw. Yeah, remember, children, we saw it in the other way. We went to the previous slide and we saw in the longitudinal way about how this line of dehiscence was. Many concepts done today. Then even stomium is one related with this pollen grains breakup itself, but still along the length which extends line of dehiscence, this groove is not at all related to anther. But now we are sorted, children. People who got this right, congratulations. If you were not able to get it right, you have some voids in your studying. So go back to the video, watch it again so that you will be clear about it, and then you can come and then you will be sorted. Yeah, this was it from my end for today's session. Keep revising, keep studying, stay home. In these tough times, you must be always, you must be safest, I would say, since the second wave is proving to be very dangerous compared to the first wave. So thank you for attending my session. So any reviews, any suggestions, any feedbacks are most welcome from your side. And then finally, this is Darshan S. Gowda signing off, and stay home, stay safe, keep studying. Bye bye.